Hi, I'm Stuart Warboys. I'm a botanist with the Australian Tropical Herbarium. It's a research joint venture based here at James Cook University Cairns campus, right in the middle of the wet tropics rainforests. Today we're going to look at using our online resource, the Australian Tropical Rainforest Plants Key, to identify some rainforest plants. In this video, I'm going to show you how to use the 7th edition of the Australian Tropical Rainforest Key, a free online resource developed here at the Australian Tropical Herbarium. And we're going to go out into the rainforest and collect some plants. Then I'm going to introduce you to plant identification using the basics of plant identification with using that key. Referring back to the last video, this is just a quick reminder that the Australian Tropical Rainforest Key was built using the Lucid system. If you're comfortable with using the Rainforest Key, then you can use one of the many dozens of other keys which are freely available online via the Lucid Central homepage. You'll come up with the homepage with the beautiful Bubbia semiocarpoides flower right on the front cover. Now we're going to start on the start identification page. So, the identification page opens up with 4 separate panels and the button bar across the top. The panels are labelled features available, features chosen, entities remaining, and entities discarded. So we'll start off in the top left corner at features available. These are the 165 features which you will use to help you identify your plant. As you choose these features, one by one, they appear in the features chosen box down below. On the other side, you have the entities remaining. As you choose features, all of the entities that share the features that you have chosen remain in this entities remaining, and the ones that don't match your data set are discarded. For example, let me just go to vines. So here I select a feature which the plant is a vine. It retains 560 entities which are vines and discards 2,441 other species which aren't vines. They're either shrubs, trees, or herbs. Now, a quick note on the entities remaining. That number includes both the families, subspecies, and varieties. So if I open up Acanthaceae here, I can see that I've got 5 species of Thunbergia plus 1 family, the Acanthaceae. So the entities remaining count will include all of the family names as well. Okay, next we'll have a quick look at the button bar. This is the buttons across the top. We'll start off over the left here. These 2 arrows are the restart button. So when you have given up on your plant and you need to go back and start again, that's the button you hit. Or if you come across a new plant and you want to start identifying that one, then you restart. Now, there really aren't too many other buttons which we'll use in this video. There is one other important button, and that's the subsets button, this one here. So if I click that, it opens up a little checkbox in which I can select a subset of the 165 characters to use. So if I only have a leafy specimen, I can select just leaf characters because I'm not interested in the flowers, the fruits, and the tree bark. Select that, and suddenly the features available drops down to 35. So that's only 35 things I have to think about, 35 characters I have to consider. All the others are just gone. That's all we really need to consider for the moment. The next step is to actually go out into the rainforest and collect some plants. So let's do that. So here we are on the edge of the rainforest on James Cook University Cairns campus. These patches of rainforest contain quite a diversity of trees. Now I can walk into the forest here behind me. It's a little bit hard to get to, but I can push my way through. But there's probably no point, because on these edges the branches hang down quite low and they're very nice and easily accessible. Under the canopy of the forest I'd have to reach up maybe 15, 20, even 25 metres to sample the canopy. But here I can just get some low-hanging fruit. So that's one sample. It's always good to have a second sample, because during the process of identification we'll pull this apart, we'll destroy it. So let's just get another piece. Always better have too much than not enough. So that's our sample, but there are a few other things we need to record, so let's go and have a look at those things. So I've just collected these samples which we're going to identify. I've got some nice leafy samples here, but there are several other features that we can record whilst we're out here in the bush. And I'm going to take you into the forest here and we're going to have a look at those features on lots of different species, and we're going to take notes on those and use that information to help us identify our plants. So let's go into the rainforest. Well, here we are back in the rainforest on James Cook University Cairns campus, and I'm going to collect a plant to identify. This one here, which I've found, will be an interesting one to start off with. So now we've got a good leafy specimen and some fruits, and we have enough material to identify this plant. Okay, so we've collected our specimen and we'll be collecting a few more, so I need to label these so we can distinguish them later on in the, when we get back to the lab. So every single specimen that you collect needs a unique number, and that refers back to the field notes that we'll be taking. So I'll show you a couple of different ways of labelling plants. First and the easiest is simply to write on the leaf. So it's the 26th today, so I've written today's date, and I've called this specimen A. Okay, so we can also use some flagging tape. And attach that to the specimen. Now finally, what the real botanists do is they use jeweler's tags. Now you don't have to use these, just the texture on the leaf is perfectly fine. But most specimens in rainforest, most specimens that are collected have these jeweler's tags attached to them. So I now have 3 separate samples from the same tree, and they're all labelled with the same unique number. That's specimen A, which was collected on the 26th. Now I can use any particular numbering sequence that I like. However, that number has to be unique, and it has to refer back to the field notes. Okay, so we've labelled our specimens, and the next thing we need to do is to have a look at some of the features which are only present on we can only see on fresh material. If we take this back to the lab, it may dry out a little bit, and some of the features we need to record now will not be visible. So let's do it here in the rainforest, and we'll put these details into our field notes. First thing to do is to look for sap. Is this a sappy plant? So what I do for that is I break off the leaf, and here I've got my trusty hand lens, and I check, is it bleeding sap? Yes, it is. It is bleeding a very slightly milky sap. The next thing I need to discuss is the presence of oil dots within the leaf. Now, oil dots are just what it sounds like. They're globs of oil which occur. They're cells full of oil, vesicles full of oil, which occur within the tissue of the leaf. So they're not visible by looking at the surface of the leaf. I actually have to see them with transmitted light. So I hold up the leaf to the light and get out my hand lens and very carefully check. Can I see small, round or sausage-shaped dots in this? Nothing visible there. Now I have to check both sides of the leaf. So they're visible from the undersurface of the leaf, but not from the top. So I can say now that oil dots are present in this leaf. So here's what the oil dots look like under magnification. Don't forget to check both sides of the leaf. You can see lots of transparent, regularly arranged clear dots, usually of pretty even size and shape. In a few rainforest species, oil dots can be coloured or long and thin instead of round. Now with the oil dots, there's something to watch out for. As leaves get older, they get affected by disease or get damaged by the slings and arrows of misfortune, and that leaves clear patches in the leaf, like this. So the next thing to look at is surface glands. So these are structures which are not embedded in the leaf tissue, like the oil dots are. They are on the undersurface of the leaf, so you can see them by reflected light, not by transmitted light. And they're really variable in shape and colour. One example of leaf glands is the extrafloral nectaries on this juvenile candle nut here. So if we have a look at the leaf of a candle nut, the leaves are quite large, as you can see, and at the base of each leaf is a pair of tiny glands. These tiny glands exude nectar, sugary liquid, and ants are attracted to that sugary liquid and they feed from that. They will fight away any insects that come to attack these leaves and protect their food source. So this is a particular advantage to this particular leaf. Now, another feature of this leaf is when you break it off, it actually bleeds sap. So lots of pale, watery sap comes out of this leaf. Something else to put in the field notes if you're collecting this specimen. The next type of leaf gland, you'll actually need a hand lens to see. This is a leaf of Mellitus philippensis. Its leaves are covered on the undersurface with tiny, tiny little red glands, which you can see only with a hand lens. And there's a relative of Mellitus philippensis, which is called Macaranga polyidinos. And its leaf undersurface is covered in much the same way with tiny, tiny yellow or clear glands. There's one final thing I want to check out while we're out here in the field, and that's the smell of the leaf. Lots of leaves have very distinctive smells, and the best way to capture that is to pull off a young, fully developed leaf, not an old one, but a young, fully developed leaf, and you see quite a few people look for the smell with their leaf, but just by breaking it in half like that and having a smell, that's not the way to do it. The way you've got to do it is actually crush it up in your hand and capture those volatiles in your cupped hands. There are some plant species, for example, your laurels, in which the volatiles really quickly disappear, and you only catch a very quick whiff of them before the leaf, before they drift away, and the leaf just smells like cut lawn clippings. So have a smell of the leaf and put that down in the notes. That's going to be a big help in helping you remember some of the plant species out there. Mangoes smell like mango. Lemons smell like the crushed leaves of a lemon will smell like lemons. And while I'm at it, there's another point. Some people will associate the presence of oil dots with the leaf having a smell. That's not always true. So mangoes don't have oil dots, but they have a very strong smell. Mercenes have lots and lots of oil dots, and they don't have a smell. Nor does this fig that I've just looked at. Lots and lots of oil dots present in this, and just smells like freshly cut lawn clippings. Okay, so now I've noted all those details on the fresh material whilst I'm in the field, so now just quickly write them down. Okay, so I'm back at the tree that I collected the sample from, and we're going to have a look at some bark features now. Now, one of the things some of the things that are easily looked at whilst we're in the field are the sap that may come from the bark and the bark texture. So the first thing we're going to look at is the bark sap. Now we need to wait around about 60 seconds because tree bark can be quite slow to bleed. But this is a fast one. You'll see pretty much straight away. So it's very slow, very meagre, but it is present. So we call this bark exudate. And bark exudate can be clear, it can be yellow, it can be red, or in most trees it's actually absent. But it's an interesting and rare character, and we put that in our field notes, and we use that to help us with identifying the tree. Okay, so there's one more feature about this tree which is quite unusual, and it's this structure here. This branch is naturally hanging down because it's actually a fruiting branch. Now I've picked off some figs from it, and here they are. These figs have appeared from this branch quite close to the ground where they're accessible to fruit-eating mammals, and when they ripen they'll be eaten by rainforest kangaroos or pigs that might happen to wander through, even cassowaries. So this feature of flowering and fruiting from the trunk is called cauliflory. It's quite a rare character, and it's a very, very powerful one for identifying plants in the rainforest. Only about 2% of our rainforest trees are cauliflorous. There are over 1,000 tree species in the rainforest of tropical North Queensland, and during the days of rainforest logging, over 150 species of those were harvested for timber. So there was, with all the identifying features of the tree, 20 metres or more, up there in the canopy, foresters often had to rely on bark characters to identify their trees. So here we're going to use some of those forestry bark characters to help us identify our trees. Now the first one we're going to look at is a fissured bark type. This is this one right here. I'll show you a couple more in a little while. So this fissured bark has longitudinal ridges with deep valleys in between them. And this is actually quite a corky texture, but we can ignore that. That's not relevant to our identification here. We have long, deep fissures in this bark. So this is a fissured bark type. If we were collecting this plant, we'd note that. Okay, another example of fissured bark is this one here. This is actually one of the rainforest acacias, and you can see once again the deep longitudinal fissures within the bark. Another bark type in the rainforest community that we're looking for is flaky bark. This one here is a perfect example of flaky bark. Very simple. The bark comes off in flakes. Watch out for large spiders underneath the bark. And those flakes break like a biscuit. They're not flexible like papery bark, they break like a biscuit. When you're trying to characterise or describe bark types, not all trees will fit neatly into the pigeonholes that we've got in the rainforest key. So this tree here has a bark which peels off quite neatly. It seems papery, but it also breaks into pieces. So this bark type here would be we would consider this both papery and flaky, or if you were unsure, you would leave the character out when you're looking at the rainforest key. Another bark type that we use in the rainforest key is tessellated bark. Tessellated bark is quite rare, so it's a very good character to note down. You'll see on this this tree here, this is a Terminalia microcarpa, which was discussed earlier in the video. It has quite well-defined little squares of bark. It's got a thick bark, and these squares are like tiles. This is called a tessellated bark type. This tree also has another distinctive trunk feature, which is the well-developed buttresses at the base. So Terminalia microcarpa is a very common tree in the lowland rainforests in northern Australia, and is quite distinctive and quite cyclone resistant. Another bark type, which is actually quite rare in the rainforest, is found on this tree right here. Now this tree is a eucalypt, Eucalyptus teraticornis, forest red gum, and like many eucalypts, it has a smooth bark. If I were describing it as a eucalypt, I would call it a gum bark, but for the purpose of the rainforest key, this is a smooth bark type. It doesn't have any fissuring at all. It doesn't have any tessellations or paperiness, so quite smooth. However, it does have flakes. Once again, flakes that break like a biscuit. One of the rarer bark types in the rainforest is papery bark. This is actually quite a familiar bark type for those of you that live along the coast. This particular tree here is paperbark, Melaleuca dealbata, and its bark type is very, very distinctive and very, very papery. So it's flexible, peels off in thin sheets, and those thin sheets are quite flexible and papery. They're not brittle like a flaky bark would be. So I could wrap my freshly caught fish in this and cook it in the fire if I wanted to. Now we've considered papery bark, we've considered flaky bark, we've considered tessellated bark and smooth bark. Now we have one more, and fissured. We have one more to look at, and this is the single most common bark type you'll encounter in the rainforest. It's nondescript bark. So this is a perfect example. It's bark that's not deeply fissured enough to be called fissured. It's not flaky enough to be called flaky. It's not in any way papery. It's just boring, nondescript bark. Right here we've got a really spectacular example of tree buttresses. This tree, it's only 30 to 40 centimetres diameter, but it has really well developed plank buttresses coming out from the base of the tree. So in this case, this particular buttress reaches to about 2 metres above ground level, and this buttress down here is relatively small. This is also a really good example of the difference in texture of the bark which occurs on the buttresses and on the trunk of the tree. So if you're ever recording bark characters, you never record bark characters on the buttress. You always use the trunk of the tree above where the buttress is attached. So in this case, the bark is quite fissured, quite distinctly fissured. In contrast, the bark on the buttresses is nondescript. Another trunk feature to note whilst we're in the field is trunk fluting. Fluting of the trunk is these deep furrows which run quite a long way up the bole of the tree. In forestry days, fluted trunks were not desirable. All of this fluted trunk was difficult to mill, and there was a lot of wastage. So foresters had to note this on tree trunks. However, that's not an issue for us. What we're interested in is deep furrows on the trunk, which are not part of the buttresses. They run the length of the trunk, and this is quite well-developed furrowing on the trunk here. This is a fluted tree trunk. Another feature of interest for foresters, which was incorporated into the rainforest key, is the presence of coppice shoots. Now, for the purpose of the rainforest key, coppice shoots are green leafy shoots which appear from the base of the trunk of the tree, or in the lower sections of the trunk of the tree. So here's a really good example of that. We've got a green leafy shoot which is arising from near the base of the tree, and it will eventually produce a subsidiary trunk. Now, there's a word of caution with coppice shoots. Coppice shoots are a really good way of sampling leaves if the tree, they're a really good way of getting a leafy sample if the tree is 20 metres high. But you do have to take care. These are leaves that are produced in the shade. Shade leaves are often much larger and have a different shape to the leaves which you'll get in the canopy. This tree, interestingly, also has aerial roots. Not a useful feature in the rainforest key, but it's unusual. And it also has flaky bark. Now, so we've reviewed bark features. Now, if I hark back to the fig that we started off with, I can't actually use the bark features in that tree because it's too small. Bark features don't develop until the tree is roughly 10 centimetres, maybe even more in diameter, whereas that tree was perhaps 8 centimetres, not big enough to record bark features. So we only record bark features on big trees. So we're on our way back to identify these plants I collected a little bit earlier. Now, whilst we've been in the bush, we've been looking at a few different features, and earlier I went through and collected these features for for this plant sample. So let me quickly review the information that we've collected whilst we're out here in the field. So, with regard to leaves, we've collected information on leaf sap. We've collected information on the presence or absence of oil dots and the presence or absence of leaf glands. We've had a close look at the bark texture. We've checked the trunk for trunk fluting, and we've checked for coppices, and we've also looked for buttresses. So these are information which go into our field notes and will be very useful back in the lab for identification. Okay, so we've been out into the rainforest and we've collected our plant, and here it is. It's a nice big rainforest plant. So I've set aside I've collected two pieces of this plant, one for preservation as part of a herbarium, and I've got a second bit here for identification purposes. So now we're going to run through the rainforest key and identify this plant. Okay, so we need a couple of pieces of equipment for our identification. Probably the most important piece. This is why I have it attached to my body by a piece of string, is the hand lens. All you need is a 10 by magnification hand lens that will facilitate looking at a few of the features. You need a ruler for measuring leaf dimensions, and you need the field notes and pencil that you took when you're out in the bush. Okay, so earlier on I pointed out that it's possible to reduce the number of features that you use in the rainforest key by using the subsets. So what I'm going to start off by doing is doing that. Now, what I'm going to select is not just the leaf character subset. I'm going to use the frequently used subset. That includes a lot of characters that I want to talk about in this video. So we're going to reduce the number of features available using the frequently used character set. As you can see, That reduces the number of features available to 54, and that makes life a lot easier. Now what we're going to do is work our way through the characters that we have available. So I start off with geographic area. I've got a selection of 5 options for geographic area. Now if you're not sure if which geographic area applies to you, you can open up a map. Now I'm based in Cairns right now, and I can see that I'm right at the centre of the geographic area Northeast Queensland. Now I get to these, I can examine maps by simply clicking on these little help buttons here. So not only do you have help outside the key, but you have help inside the key. Just a quick note on the geographic features. The Cape York Peninsula side part of the key only applies to plants north of Princess Charlotte Bay. So if you're in Cooktown, for instance, and you collect a plant, you're not on Cape York Peninsula. You're actually still in northeast Queensland for the purposes of the key. Let's close that down, and we'll move on to the next feature that we know something about. And that's, well, we don't know what the family of this plant. This is a mystery plant for us. We don't know what the family is, and there's about 197 families to choose from. So let's skip that family thing, move on to habit. Is our plant an epiphyte, a grass, a herb, a mistletoe, a palm, a shrub, a tree, or a vine? And obviously it's a tree. It's a plant which typically flowers when it's greater than 6 metres tall. Now there's a bit of a glitch in the key here. If I look to other plant features, there's nothing there, so you can skip that. Leaf features. So now we're going to work our way through the leaf features as we identify this plant. Look at this plant. The first thing to consider when we're looking at leaves is whether the leaf is simple or compound. A simple leaf comprises just a simple leaf blade coming off the stem. Looks like that. A compound leaf is a structure which comprises many leaflets. The difference between simple and compound leaves is something that actually trips up a lot of people. So we're going to actually have to dig a bit deeper here. So let's have a close look at our plant. In flowering plants, a leaf arises from a point on the stem called the node. Okay, so a stem is a woody, usually cylindrical portion of the plant which ends in a terminal bud. And here I can see there's a terminal bud because it's producing new leaves. Now, right here I have a compound leaf coming off the stem, and that arises at a point on the stem called a node. Now I know this is a node because right here I have a little knob of meristematic tissue called a bud. So this bud has the potential to grow into a bunch of flowers or a new branch. So there is always a bud in the angle between the leaf stem and the leaf petiole and the stem itself. So this structure here, this tiny little knob of tissue, is the bud. Everything beyond that is the leaf. So if I break off this structure at the bud, I end up with a structure which comprises many leaflets. This is a compound leaf. So the first thing that I enter is a compound leaf. Now, the next thing I need to consider is the leaf arrangement along the stem. And that's a pretty easy one to do. Leaf arrangement along the stem gives me 4 options. Firstly, alternate. Secondly, opposite. Thirdly, whorled. Or fourthly, basal or radical. Let's unpack that a little bit. If I go back to that node situation, node structure I mentioned earlier, here I have a node where a compound leaf comes off. If I have just one leaf coming off at that node, it's called alternate. And if I have more than one leaf coming off, if I have 2 leaves coming off of that at that node, that pair of leaves is called opposite. And if I have 3 or more leaves coming off at that node, I have it's called whorled. I have an example of a whorled leaf right here. So I'm just going to trim the stem back a bit so to make my point clearer. Here I have a node right here with 4 compound leaves arising from that node. So that's a whorled leaf arrangement. This is actually Alstonia. It's a relation of the frangipani. Okay, back to it. So we've worked out our plant of interest has an alternate or spirally arranged leaf arrangement. So I select L5, alternate or spirally arranged. Keep on working through things. My plant has stipules present. What are stipules, I hear you say? So stipules are outgrowths of the stem which protect the young, delicate, developing bud. So you'll often find these in figs and relatives of figs. You'll often find them in this plant species. In this plant, they're actually quite small, so I actually need to get the hand lens out and look for the scars where the stipules have fallen off. So they're quite small on here, but they are present. Moving on to the next feature. Oil dots. Now, when we were in the field, we had a close look at this plant, and we determined that it didn't have oil dots. So I shall select oil dot character L13, oil dots absent. Now notice we've only selected 5 characters, and we're already down to 99 entities remaining. So we're doing pretty well. Let's continue. Leaf length breadth ratio. Now, I told you earlier that this structure here is the leaf of a compound leaf. However, for the purposes of the key, we're not going to measure the leaf length breadth ratio in this whole structure. We're actually going to pull off a single leaflet. Now, when we pull off a leaflet, we're not going to take off the largest one. We're not going to take off the smallest one. We always select the average leaf. So, looking at the specimen, I'd say this one here is an average size leaflet. Let's measure it. We're interested in the length breadth ratio of the leaf blade. So I'm not measuring this structure here, the leaf stem. I'm actually just measuring the big flat bit, the leaf blade itself. So it's 16 centimetres long by 8 centimetres wide. Leaf length breadth ratio is 2. Nice and easy. So my leaf L15 is the correct answer for this plant. Leaf margin. So once again, we have a look at, this requires a close look at the leaf. Now, I can see on close examination of this leaf that it's finely toothed. Let's have a quick look at the help pictures on the in the key, just to clarify things. If the leaf margin is smooth, there are no indentations or serrations or crenulations which are visible to the naked eye. The example pictures here show just 2 leaves which are, have an entire smooth leaf margin. Leaf margin toothed. You can have either sharp, pointy teeth along the leaf margin, or you can have curved, rounded teeth on the leaf margin. Either will be called toothed leaf margins. Now, sometimes with older leaves, they get attacked by insects, or they get damaged, they get injured, and bits of the leaf are broken off. So the margin in older leaves can look like it's toothed. That's why you always have to pick a nice, fresh, young, fully formed leaf because it has all the characteristics you're looking for intact. So L18, this leaf has a toothed margin, and look at our entities remaining. There's only 20 left. We're doing well. Intramarginal vein. An intramarginal vein, let me show you a picture of an intramarginal vein. An intramarginal vein is very common in the family Myrtaceae. You'll see it on gum leaves and you'll see it in lily pillies. It's a vein which is usually quite fine, and it runs just a millimetre or two inside the leaf margin. And you can see here this leaf, which is actually a lily pilly leaf, shows it quite well. You'll find intramarginal vein in some grevilleas as well. So, if I have a close look at my leaf, an intramarginal vein is definitely not present. It's absent. Select L21. Lateral vein angle. Okay, this question asks, what's the angle of the secondary veins in respect to the mid vein? Okay, so this structure here running up the middle of the leaf is the midrib. Branching off of that at regular intervals are secondary veins. So what is the angle of those secondary veins in relation to the midrib? In this case, that angle is very close to a right angle. So I can say that lateral vein angle is more than 60 degrees. Very close, it's actually very close to 90 degrees. L34. Undersurface stomatia. Okay, so stomatia are little pockets on the back of the leaf, usually where the secondary veins meet the midrib. You might have a dozen stomatia on a leaf, or you might have just one. Or maybe some leaves on a plant will have them and others won't. You'll need a hand lens and have a close look. And if you're going to find a stomatia on the back of your leaf, it's going to be where the secondary vein meets the midrib. So let's check out some of those. Now, in this case, there are no stomatia at present, so I just skip that character. Move on to the next one. Next character questions, is the undersurface smooth or is it hairy or sandpapery? Now, if you can't see hairs, sometimes you just need to rub the leaf against your lips. Don't do that if it's a stinging tree. That could be disastrous. If you're unhappy with that, check for hairs with a hand lens. In this case, I'm uncertain. I'm actually not sure whether there's hairs on the back of this leaf or not. So what I'm going to do is I'm going to skip this character. And here's the first instance of the most important thing to remember about this lesson. If in doubt, leave it out. Just skip it and move it on. Move on to the next. Undersurface colour. Okay, so this is a nice easy one. Nice easy character to use. And you always use fresh leaves to do this. So the way you compare the colour of the top surface of the leaf with the bottom surface of the leaf is just fold the leaf over, like that. And most rainforest trees, they're paler on the undersurface than they are on top. Now if you have a gum tree or a wattle, more often than not they'll be the same colour on both sides. So I select the character paler green than the upper surface, L39. So now we're going to look at the length of the petiole on the leaflet. Technically it's called the petiolule, but that's irrelevant to this story here. So what I need to do is measure the length of the petiole, which is 3 centimetres, and length of the leaf, is 14 centimetres. So the petiole length is very slightly more than 1/5 of the leaf blade. So I select the petiole is long. So the next feature to consider is the petiole winged or not. Now, petiole winged is extremely rare. If you ever used kaffir lime leaves in cooking, you'll see that that is a really good example of a winged petiole. The double leaf structure in a kaffir lime leaf is actually a winged petiole plus the true leaf of the plant. So here's another example. This picture here shows a leaf blade with a petiole, and this red highlighted structure is the wing on the petiole. Our petiole is definitely not winged. So we just skip on to the next character. Pulvinus. Now, pulvinus is a swelling where the leaf blade meets the petiole. So, to be technically correct, it has to be in contact with the leaf blade. Let me show you the picture here. You can see the red structure indicated. The pulvinus is indicated here with the red arrow, and it shows up as a slight swelling. It's going to be quite difficult to see, but right here is a swelling at the top of the petiole. I can actually feel it. In some cases, actually easier to feel than it is to see the pulvinus. The pulvinus is definitely present on this plant, so I'm going to select that. Now, look over at the entities remaining. I'm down to 9 species. So I could stop here, but I just want to have a quick look at 2 more features, and then we'll have a look at the entities that are remaining. Firstly, compound leaf type. This asks you what type of compound leaf it is. So L48 is compound with 3 leaflets. So let's have a quick look at that. So compound with 3 leaflets means that the plant consistently always has leaves comprising 3 leaflets. Some plants with compound leaves, that number of leaflets varies from 2 to 5 to 7. What you have to have is a plant where the number of leaflets is consistently 3. Examples of those are the pink euodia or many of your forage legumes. Palmate digitate means a leaf form where a compound leaf form where all the leaflets arise from one central point, much as illustrated here. A pinnate leaf form has the leaflets arising either side of one single central axis. The axis is unbranched, which is different to a bipinnate leaf form. This is the bipinnate leaf form here. You can see that it has a central axis, and secondary axis is coming off of this central axis, and so actually the leaflets are actually attached to these secondary axes. So this is a bipinnate leaf. It has leaflets coming off the secondary axes. If the leaflets were coming off the tertiary axes, then it would be a tripinnate leaf. Going back to our original leaf, this is a pinnate leaf. Now there's one more thing to have a quick look at, and that is the terminal leaflet present or absent? So let's have a look. I'm going to run up the central axis of the leaf, and does this central axis end in a leaf or is it not? In this case, it definitely ends in a leaf. So a terminal leaflet is present. So now we're down to 5 entities remaining. So I'm not going to take this any further. I don't need to. As soon as I get below 10 entities remaining, I can start looking at the pictures in these entities. So let's work through those entities remaining. So here I can review, I have two options. I can select this little coloured box, which shows me pictures, or I can select this little textbook, which shows me the full fact sheet, which includes the pictures. So let me show you the pictures here. So here we go. The leaf pattern, the vein patterns in these leaf X-rays are very valuable for identification. So I'm going to work through, just quickly scan these. You'll notice that there's 5 entities remaining, but there's only 3 species and 2 families. Next one. Is Garuga floribunda. Next one is Dinospoma erythrococcum. Now if I spent the time to look closely at this, I would find that the best match for the species that I've got in front of me is Herbarium australianum. Always double check yourself by reading through the full list of characters. Don't just rely on pictures, read through the characters as well. That's a really important thing to do. So we've managed to key this plant out to a correct identification. I'm satisfied I've reviewed the notes, I've looked at the pictures, and I'm satisfied that this species is Herbarium australianum. Not all plants are that easy, and quite often you can work your way through all the visible characters on a plant, and you'll end up with just maybe a 30 or 40 to choose from. So know that you will not be able to identify all the plants you collect. It can be a bit irritating, but that's just a fact of life. With many plants, you need full flowers or full fruits in order to effectively identify them. Okay, so now that you've stuck around until the end, I'm going to share with you the 4 secrets to identification of plants using a lucid key. These secrets don't apply just to the rainforest key, they apply to all lucid keys. Firstly, practice with what you know. Key out plants that you already know. Key out something familiar like Lantana camara or bottlebrush or flame tree, the Brachychiton cerifolius. If you know a plant, if you're certain of the identity and in particular the scientific name of it, and you run through the key to identify that plant, you'll learn very quickly if you're going wrong, where you're going wrong, and why you're going wrong. The next secret is start off with the big features. What we've just done is a simple training run through of the features of the key. However, when you come to know your way around the key a little bit better, start off by choosing 3 or 4 obvious features. So let's have a look at our Herbarium. If here's a flowering specimen of the Herbarium. If I was keying this out, the first things I would see are A, the compound leaves, B, the white flowers, and C, the fact that the flowers are in large panicles. So they are 3 obvious, safe, easy characters. Always use safe, easy characters to start off and narrow down the number of entities remaining. Then start working through the features step by step. Third secret is use the best button. Let me show you what the best button is. The best button is actually this little magic wand, this blue magic wand at the top of the screen. If I hit that, what that does is selects the best character to reduce the number of entities remaining. And that's really what I want to do. So if I hit it here, it will ask me to count the number of sepals on the flower. Now, quite often when I use the best character, the magic wand character, it comes up with characters that I can't answer. On my specimen, I have no flowers, so I can't answer questions about the number of sepals in my flower. So what I do is I just keep on hitting best until I come up with a character that I can use. Keep on hitting, keep on hitting, keep on hitting. In this case, it's asking me about the leaf base shape. And if I want to do a look at the leaf base shape, I'd use the help notes to help explain those different terms to me. Now, the process of selecting 3 or 4 safe, obvious, easy characters, followed by using the best button, is probably the best way to identify plants using any lucid key. Now, the final and most important feature is, if in doubt, leave it out. If you don't know a character, if you're finding yourself having to think about a plant feature in order to come to an answer, if you have to think about it, just skip it and move on to the next easy character. Always stick with the easy characters first. And that way there's less chance of you making mistakes. In the same way that I was uncertain whether this plant had a hairy leaf undersurface, I just skipped it and moved on to the next one. And I still came to a right answer for this identifying this plant. Thank you for watching. I'd like to thank Frank Zich for his input, Darren Crane and Susan Lawrence for facilitating production, and Teresa Stevens for her help in making this video. And this is Stuart Warboys from the Australian Tropical Herbarium, signing out.